Okay, now we're going to talk about one of the simplest but also another very useful method that is used in the lab, and this is called gel electrophoresis. All right, so gel electrophoresis, and the concept here is very simple. Electrophoresis. It's kind of a weird word. Gel electrophoresis, and as you might imagine, this involves a gel and it involves applying electricity to that gel to separate different things based on their size. So basically, the key idea for gel electrophoresis is that if you have two different particles, let's say you've got this guy here, this big blob, this red blob, and you've got, say, this tinier little guy, this blue blob, and if you send them through a maze of a gel, and I'm gonna kind of explain why it's a maze. If you send them through a gel, then this little blue guy, he's gonna go quite far quite quickly. And if you take this red guy, he's gonna go more slowly. Okay, and he's gonna go more slowly, not because he's bigger and heavier, but because as he tries to push himself through the gel, he kind of bumps into things more quickly, and so he sort of stumbles through the cross-linking of the gel, and I'll talk about what that means a little more slowly than the blue guy. So the key thing here to understand is just to understand that a gel is a bunch of sort of cross-linked, oops, well, okay, ignore that little red line, is a bunch of little molecules that are cross-linked to each other. So when you have a gel, you have kind of a maze, of a mesh of particles. You have all these little weird cross links, but you have these holes, right? So you'll notice that there are these nice hole areas right here, like this and this. And these holes can be different sizes, right? So you might make a different gel that has bigger holes like this, maybe like that. And you see here that you have this much larger space to fit through. But the basic idea here is that then, when you have Mr. Red Molecule coming through here, he's going to kind of have to wedge his way into one of these holes. Maybe he's going to have to like bump around for a little while until he finds a big hole over here, and then he can finally get through. Whereas the blue guy, he's gonna come along, he's gonna cruise by, and he's just gonna go right through the hole. He's just gonna go right through. So that's the main idea. And the way that we actually do this, the way that we force the small molecule and the big molecule to move, kind of in one direction, the kind of force that we apply to the system that keeps things moving in this direction, the force, comes from an electrical field, right? And so this is pretty simple. So you just do this by having a plus end down here and a minus end up here. And if the particle that you're interested in is negatively charged, right? Let's say this is negatively charged, then it's going to go towards the cathode. It's going to go towards the positive end, and then that's going to pull it through the gel at some speed. So the molecules that we're actually interested in are perhaps the molecules that we talked about in the last video, PCR products, right, which are either DNA or RNA. Actually, just DNA. You can't PCR RNA. So it could be DNA products. You can also use other things, like you can use gel electrophoresis on proteins, although it's a different kind of gel because proteins are a different size than DNA. Remember, DNA is kind of like this long, snaky thing, so it actually can get through the holes pretty easily, although bigger bits of DNA still move a little slower through the gel. So then, for example, let me just draw that out. So this should be like a long bit of DNA, and then your blue example would be a little short bit of DNA. And then this would tend to move faster than this through a gel. And just trust me on that. I know it seems a little weird. It seems like they would both sort of snake through at the same rate, but it just ends up not being how it works. So we can do this in the lab, and we do this very frequently. And I'm going to show you kind of the system that is used for that. The system is to have a little box. I'm going to show you in profile. You have a little box like this, a little flat end, and it has two troughs, right? So you have a trough over here. Actually, the water fills the whole thing. So you have water here or actually it's a buffer, right? So it's at some set pH. You have water over here, but at some set pH. Remember, that's important. It's buffered. And then you put a little slab of gel right in here. And this is an agarose gel usually, so I'm going to write that. Agarose gel in the middle. And then you have a positive charge over here. You have a negative charge over here, and we do that by having these little electrodes that come off, and they go to a little box that's plugged into the wall, and it generates a constant current, which is very important. And we then add our DNA to this end of the agarose gel, and it goes towards the positive end. It slowly but surely migrates towards the positive end of the gel. And if you sort of take this and you kind of flip it you sort of flip it around so that you're looking from the top down now. So let's look from this perspective at this thing. If you're looking from this perspective, what you see is a gel. You have a trough of water up here. You have a trough of water down here. The gel that we actually put in there has these little wells that are pre-made into it, and we make those wells. But anyway, these little wells that we're going to put our DNA into. And then actually let me make some of them blue, right? So let's say this one is a smaller molecule than this one. And then when we have the current run, right, this is the plus end and then the minus end, the blue one is going to move fast, right? So the blue one's the small DNA fragment. It's going to go like all the way down here, right? These two guys are going to run all the way down here. And the red ones, they're going to run up here. They're going to migrate much less far because they're bigger, and so they bump into the crosslinks more, and they can't shove their way through the holes. But what's really kind of fun about agarose is, let me just extend this a little bit. So let's say I'm going to add one more lane. These are called a lane of the gel, each sort of individual slot that the sample runs in, each kind of vertical column. So I'm going to add one more column. But what's really kind of fun is if you have the blue, the small molecule, and the red molecule in the same well, right? Let's say you include both of them in the same pool of DNA that goes into this electropheresis procedure. Then the red one will migrate slowly, and the blue one will migrate quickly. And then, kind of cool, you have this separation. You've actually separated the two molecules by size. And you can then do things like cut this little bit of the gel out, and you can purify this little shorter fragment out from that. And I'll talk about how you do that in another video. But this little guy right here, you could take this guy, you could cut him out, and you just do this using a razor. I mean, it's like the simplest thing ever. And you take it out, and you can extract, the red bit, so that then at the end you have a little container. Maybe you have like a little tube, and you have only red bit in it. Where before you had red and blue, you have like a little tube. It's all red pieces of DNA. And this is useful sometimes for certain different situations, and that'll become increasingly obvious as we talk about the kinds of things you might want to try and isolate and separate and blah, 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 and dot, dot, dot. But the key here, gel electropheresis, it tells you what size something is. You can use it to separate bits of differently sized DNA, and it's really quite easy to do, and you will probably be able to do it on your first day in the lab without any trouble at all. And that is the kind of science I like doing. So that is that video. See you later.